as you can see, I've changed the title of my presentation, which now reads Development of a New Rapid Prototyping Strategy for the 3D Fabrication and Biofabrication Tissue Engineering. So tissue engineering is a modern and multidisciplinary research field which aims at restoring tissue and organs whose functionality has been compromised by diseases or trauma. A common approach followed in tissue engineering practice is the seeding of cells on a supporting structure, which is commonly called the scaffold, together with bioactive cues. After a functional tissue has been obtained, is transplanted into the patient body. So essentially, tissue engineering consists of two main steps. The first one is the manufacturing of a supporting biomaterial, and the second one is the migration or post-seeding of cell in either static or dynamic environment. During the course of years, many technologies, many fabrication technologies have been developed for the manufacturing of scaffold. So scaffold characterized by either a random or ordered structure can be fabricated, or also scaffold which consist of fiber meshes. One of the most advanced recent technology allows the production of scaffold according to a predetermined design. Many materials can be, so the materials chosen for the fabrication of scaffold must be biocompatible, biodegradable, and favor cell adhesion and proliferation. It can be made either from a natural or synthetic polymer. So one of the most successful and technology used in the scaffold fabrication is rapid prototyping. So in this technology, the object that you want to fabricate is modeled at the computer using dedicated software, and then is sliced into a series of horizontal layers, which brings to the generation of a printing file sent to the rapid prototyping machine, and then the object is fabricated layer after layer. So there are the main technology used in rapid prototyping are laser-based, nozzle-based, and printer-based. So the success of rapid prototyping relies on the possibility of acquiring a detailed anatomical map of the defect by means of X-ray computed tomography and magnetic resonance imaging. Then the defect is computer modeled and realized with the rapid prototyping machine. So the deposition technology which is chosen is strongly dependent on the biomaterial being processed. And this is even more true in the case when you want to deposit at the same time simultaneously cells and scaffolding material. So what I will show in this presentation is I will illustrate you the development of a new deposition strategy, which is particularly suitable for biopolymers. And then so the rapid prototyping of scaffold made of alginate, and also the post-processing of such kind of scaffold. And finally, I will show you the rapid prototyping of scaffold and cells at the same time. So the hardware setup consists of 3 linear stages assembled in a X, Y, Z configuration. This rapid prototyping machine is computer controlled and is associated with a syringe microfluidic pump for the control of fluxes during the 3D deposition. And the deposition strategy consists of wet spinning of alginate fibers using a coaxial needle system. So here is this. There you can see the scheme of the coaxial needle system consists of an internal needle through which alginate solution is extruded, and an external an external needle through which a calcium chloride solution is extruded. Here is a photograph of the tip of the dispensing system. As you can see, the internal needle protrudes out of the external needle a little bit. So I would like to show you a video, but sorry. So the fiber diameter can be controlled. Sorry, this is. I went. I have some difficulty with this here. Okay. So the fiber diameter can be controlled by regulating the speed of deposition. So here are reported the two simple formulas. The meaning of the symbol are shown in the cartoon. So if you combine these two formulas, you can see that the fiber diameter is inversely related with the flow, with the speed of deposition. And so in this graph, you can see that how the fiber diameter decreases with the speed of deposition, and also decreases with the flow rate of the calcium chloride solution. So here I reported the SCM of 3 scaffold deposited with the coaxial needle system. So according to 3 different patterns of deposition, geometrical layouts, so square, hexagonal, and radial. And here there are 3 scaffolds characterized by the same interfiber distance, but increasing fiber dimension. So here is a 3D reconstruction of a scaffold, of a bioprinted scaffold in the swollen state obtained by computer X-ray microtomography. One of the problems of the calcium physically cross-linked scaffold is that when you put in a cell culture medium, they tend to lose their ordered structure, and for prolonged period of time, the fibers tend to dissolve. This is due to the presence of a relatively high concentration of ions in the cell medium culture, which compete with the calcium and compete in the interaction between calcium and the alginate. So what we did was to coat the scaffold with chitosan in order to form a polyelectrolyte complex. And then the scaffold were covalently cross-linked with the ADC and the genipin. And such treatment preserved the scaffold in cell culture medium for a long period of time. So the cross-linking of chitosan with the genipin gives in part to the scaffold a characteristic blue colors, as it is evidenced here in this confocal laser microscopy image. And here you can see the image of a single fiber where you can see that the chitosan penetrated inside the fiber for about 40 microns. We also characterized the mechanical properties of the scaffold according to a perpendicular and parallel direction with respect to the plane of deposition. And as you can see, the scaffold exhibits anisotropic mechanical properties. So we see that the scaffold with HEPA-RG cells, which is a differentiated human hepatocyte, so the cells adhered quite well to the scaffold and proliferated a lot. So here I report the viability, functionality, and the detoxification activity of HEPA-RG on the scaffold. So we have seen that these dispensing systems allows the deposition of a 3D structure with high fidelity and reproducibility. It's also quite easy to use and fast. We may wonder if it's possible also to print multi-material components and cells and scaffolding material simultaneously. If we assume that this is possible, a question arises regarding the bioactivity of the bioink. Alginate is known to be biocompatible, but is not cell adhesive at all. So we thought to blend alginate with the gelatin methacrylate. Gelatin methacrylate is show a strong bioactivity attributable to its abundance of integrin binding motif and metalloproteinase degradable groups, which promote cell adhesion and migration through the matrix. So we, again, in a similar fashion as shown you before, we extruded a blend of alginate, gelatin methacrylate, cells, and a radical photoinitiator through the internal needle, and the calcium chloride through the external needle. After the scaffold, after being deposited, is exposed to UV light to promote the chemical cross-linking among gelatin chains. So an important issue regards the optimization of the bioink. So there are two important requirements that the bioink needs to satisfy. The first one is the maintenance of high cell viability, and the second one regards good printability of the bioink. So we may think to keep the concentration of calcium chloride relatively high, and in this way have a good printability of the bioink, but high level of calcium may be harmful for the cell. On the contrary, we may think to decrease the calcium chloride concentration, so in this way we preserve cell viability, but the printability, we don't have any printability of the bioink. So the best compromise was represented by using an alginate relatively high concentration of calcium of alginate, while keeping the concentration of calcium low. So then we characterize the viscosity as a function of shear rate of the bioink and also of alginate and gelatin methacrylate as a reference. As you can see, the bioink exhibits almost a Newtonian behavior in most of the shear rate explored, and the viscosity is relatively low. In the usual bioprinting practice, solution characterized by high viscosity and that exhibit a high shear thinning response during extrusion are used. This to avoid that the printed strand spread out on the surface. But a high viscosity of the solution may hamper the dispersion of the homogeneous dispersion of a high concentration of cells. On the contrary, in our case, the low viscosity of the solution facilitate the dispersion of cells of a concentration of cell of the order of 10 cells, 10 to 7 cells per milliliter. So here is reported the bioprinted scaffold in the swollen stain, and here the relative SEMs. Then we optimize the bioink solvent composition to match the condition of neutral pH and isoosmolarity. So when the bioink solvent is represented by water, the cell viability is relatively low, is of about 35%. When APES was used as the bioink solvent, cell viability increased above 40%. When APES and the fetal bovine serum were used, the cell viability was around 80%. Then we decided to suspend and to print endothelial, together with the scaffolding material, endothelial cells, and we observed the level of cell viability as a function of UV exposure time. So for exposure time until 30 seconds, the cell viability was high and constant. Beyond this temporal limit, cell viability tended to decrease progressively with the UV exposure time. So the spreading behavior of HUVEC is strongly dependent on the mechanical properties. So we determined the elastic modulus as a function of UV exposure time. And of course, as you increase the UV exposure time, the degree of cross-linking among gelatin chains increase, and as a consequence, also the elastic modulus. So here I reported the spreading behavior of HUVECs. As you can see that for UV exposure time of 15 seconds, so relatively low mechanical properties, the HUVECs retains a spherical symmetry. As the exposure time and as a consequence the elastic modulus increase, cells tend to spread more and more. After 10 days of cultures, the cells, HUVECs, line up at the edge of the fiber, assuming a long-range arrangement, and formed a cell monolayer. So here you can see confocal laser microscopy images taken from the top, transversal and longitudinal direction, and you can see the formation of a tubular or endothelial network. Another interesting feature is shown in this photo. So at the correspondence of the contact area of fibers belonging to adjacent layer, there is the formation of a perpendicular network. Then we also checked that if the viability was uniform inside the scaffold, and indeed it was. And then we immunostained the scaffold, the biocontact, to see the level of expression of CD31. CD31 is a membrane protein which is highly expressed in endothelial cells. And as you can see, the strong signal throughout the scaffold, and also the location of the CD31 receptor at the periphery of the cells indicate a normal development of HUVECs. So to assess whether this biocontact could serve as a platform for in vitro cardiac tissue engineering application, we seeded the primary cardiomyocyte on top of bioprinted scaffold containing human cell. After 2 days of cultures, the cardiomyocyte started beating synchronously, as is also shown in this graph, where the beating frequency was monitored in 4 different spots, and as you can see, the profiles are very similar. So finally, to illustrate the possibility of creating a multi-component and a multicellular tissue construct, we integrated our dispensing system with a microfluidic chip. So the aim is to create, to print a scaffold formed by different material arranged in organized matter, in organized way, to replicate to some extent the heterogeneity of native tissue. We used 2 bioinks consisting of alginate and containing 2 red and green labeled fluorescent beads. And as you can see, if we sent alternatively the 2 bioinks to the extrusion system, in this way printing scaffold characterized by alternate layer of different composition. Or we may also send simultaneously the 2 bioinks to the extruder, in this way printing a composite fiber. So in conclusion, our deposition system allows the deposition of with high fidelity and reproducibility The fiber diameter range from between 150 to 100 microns. There is also a high cell viability. There's a uniform survival throughout the 3D structure. And the endothelial cell matured into a prevascular 3D network, which may be an approach for in vitro vascularization, which may represent an in vitro vascularization strategy. And also the coupling between the dispensing system and a microfluidic chip allows the preparation of heterogeneous multicellular structure. So with this, I've concluded my presentation. Thank you.